Yeah, I did that the other day. I might as well cut a case. Phone is fine, but the case broke. So I guess I can do its job.
When I first got this one, I opened it in Talbot, and the way I opened it, this phone wasn't secured in there very well.
Oh,
like
no.
the packaging that I found this phone is not very good. I don't know why. And I opened it, and before I even touched it, it was flying. Luckily, not a scratch on it. Don't know how that happened. Luckiest case of the phone ever. Really bad.
It's a Samsung Galaxy S40.
Wow, that's a lot of things.
Yeah, there's a lot of things.
Oh yeah, the Razor was not a flip phone.
You know,
It's basically the Nokia of
I'm really mad at that it's not it's not like my snap score is not increasing, like getting snapped.
Maybe case.
Yeah.
It comes out today, I believe, but I also don't know. My grade has nothing to do with $1.09. So, yeah. And it looks interesting, but not all that much is changed from this one. Yeah, so it's like, I didn't think much of time to worry about it.
That's interesting. I've never heard that we're going to get like all of discussions. My roommate yeah, I know. But like, I mean, I had bought that growing up and I'm not from Ohio.
yeah, well, we're in your heart.
Yeah, I was. But they have friendlies, which they take away all of those in there, or almost. It's so sad because I used to love them because I was a kid, and their Sundays are great. And I'm like, oh no, it's another textbook. Or like, okay, like this year I was like really bad like accident like come to school. So I like left my computer charger and my raincoat blankets at home. Yeah. And so I was like, okay, got a package. This is my raincoat. Right.
I love my raincoat at home too, but my parents
together in like 1 or 2 months.
I did not notice that.
Oh, I forgot. Okay, cool. I'll try to get this off. Where? About like where I live.
We don't have any new handouts today, but if you need the handout from the other day that had the questions for today, I've got extra copies here. Nothing new up front, though. Hi, Sarah.
You can always watch my babies.
Hi, Alex.
That's where I'm going to be doing work.
Yeah.
She's a girl, I'm going to be watching right now.
Yeah. Because she probably forgot to record it on it. She doesn't want to watch it. And I mean, like, when it does, and if I'm not home, I would like to remind her to text her several times.
Yeah, my sister's just got it. So she's like on like season 4.
Oh, okay. It's a nice big pop up.
Yeah.
I kind of want to watch the last episode of season 11 before I see. Even though they do a nice recap of every episode.
Yeah.
Away I go into S&P. See you later.
So sad.
Okay, we've got 12:30. Why don't we get started? Good afternoon, S&P. That's a terrific response to end week number 4. Who's ready for a weekend? Yeah? Okay. As I told my other class, we're going to catch up on lots of REM this weekend. It's good for your brain, it feels good, and it helps you learn. So please sleep well this weekend as much as you can. And I've got some great news. Our writing assignment is due on Wednesday. Isn't that great? Give it up for the writing assignment? Yeah? Okay. All right. So we went over all those details the last time. You had the handout, but also it's available here in Blackboard under the course documents. You'll click on that and you'll submit that. Just as a reminder, many people who are in my other class know the routine. For those of you who are less familiar, we will bring a paper copy to class just in case on that day. But you're really going to be turning in your assignment electronically by pointing on that, clicking on it. They'll give you an upload opportunity. You can upload your MS Word file. I'll provide feedback to you. I grade anonymously, and I'll provide feedback to you through Blackboard that way. But we'll have the paper just in case it doesn't work. And then maybe the second, third, and fourth writings, we can dispense with the paper entirely. Okay? All right, really good. So I'm looking forward to that. That's for Wednesday. For Monday, we have one more TED Ed coming up. Thank you all for your TED Ed responses. This group really understands how to do on-center and off-center cells. So you may have just received a response from me through TED Ed, but many people in the room answered that question in Draw 2, and you had excellent examples of on-center and off-center cells. So thank you for that. We'll have another TED Ed coming up for Monday. This will be on the eye and on the brain and seeing. So today and the last couple of days, we've been working on the human eye, doing the transduction. We talked a lot about photoreception. We talked a lot about the properties of light and so forth. Now we're going to make our way through the visual pathway up to the cortex on Monday. Okay, so that's what those questions are going to be focusing on. All right, just by way of review, you know, we like to go 2 steps forward but 1 step back, and we all try to find our blind spots. So I won't use any gesture whatsoever. Why don't we see if we can find the blind spot in your right eye? So let's see how we do there. Okay, looks like most people are getting into position. Right, so, and you can sample your thumb, right? And if it helps, maybe this will help. Why don't you try to focus on the 5 and the 92815? Why don't you put your fixation there? You can hold your thumb up there initially, and then what you can do is, while you're keeping your eye fixated on that 5 and your thumb is kind of in the way, just ever so slowly move the thumb a little bit off to the right. Okay, so and you're keeping your eye on the the 5 and the 15, right? You're keeping your eye there. But the thumb is a little bit off to the right, and at some point it will disappear into your blind spot. If you go too far off to the right, it reappears. Okay? But if it's just a few degrees off to that one side, you'll find that. How many people found their blind spot? Okay? It really helps to have that little page, because that's nicely laid out at arm's length. That works for just about everybody. So we found our blind spot. How many full moons did we say would typically fit into the blind spot? 12, okay, right? And so just to give you some of those numbers, 12, so a dozen full moons. It looks like it's this huge thing in the sky, but it's actually about half a degree of visual angle. And 1 degree of visual angle occupies about 300 micrometers on your retina. It's a point we didn't make the last time around, but it's a fun factoid to have. How big is a micrometer? Anybody help us out with that? We've talked about nanometers, right? But we haven't talked about micrometers. So they're both fractions of a meter, like a millimeter or a centimeter, right? So the micrometer, any idea about that? How about the nanometer? What did we say that was? That was a billionth, right? With the billionth of the B, and that was expressed in exponential format. 1 times 10 to the -9, right? Okay. So a micrometer is 1 times 10 to the -6, which would make it a 1 millionth. Okay. So 1 degree of visual angle is about 300 micrometers, 300 1/millionths across on your retina. Okay? And your optic disc is about 6 degrees of visual angle, so 1,800 micrometers. You can fit 12 full moons in there. A moment ago, I tried to get this little news blurb. It was only about 30 seconds long on the upcoming event for this Sunday night. Really excited about it. I think it's, and we can look up the time, but I think it's about 9 o'clock or so on Sunday night. We have the supermoon, right, which means that the moon is going to be relatively close to us in its orbit, and it's up to 17% larger than it normally is because it's that close to us when it's full. So really, really cool. And then we also have the unusual event of the lunar eclipse, and that won't happen again for many, many years, and it hasn't happened since 1983 or something like that. So we're in for a treat. I hope the sky holds up. Hope that we don't have any Ohio rain coming our way. But it might be fun to go out there and see if you can get the full moon, the supermoon, before it goes into eclipse to disappear into your blind spot. Always a good trick. And I usually get lucky during S&P in September. At some point in September, there's going to be some full moon, and I usually have my S&P students try it out if they can remember. I might be sending you all an email just saying supermoon in 1 hour or something like that. You don't have to reply to that, but that's just my reminder to you. Okay? All right. Any other questions on the blind spot? Yeah? Okay. There were a couple of TED Ed questions that we didn't quite finish up from the other day on part 2, the human eye part 2. Right? So part 1 was really talking about these very basic physics principles, and we started to get into the physiology of the eye. Then we developed that a little bit further. And at one point in the TED Ed questions, we asked about something called the principle of univariance. First, who just remembers hearing about that or remember reading about that? Okay. Sometimes students find that difficult. Would somebody like to try to get us started in the principle of univariance? Principle of univariance. Alex has something, then we'll go to Tani. Yeah.
Is it that all wavelengths across all light are lost at the point of transduction?
Oh, really nicely stated. Okay, yes, at the point of transduction, all wavelength information is lost. At the point of transduction, all wavelength information is lost. Does anybody recall how we defined information a while back? We had it up on the board. Do you remember that? The reduction of uncertainty, right? Also, Tani had a hand up. Was that what you were going to say? Okay. So at the point of transduction, and what we mean by transduction is there's some energy out here in the world. That light is shining in my eye. There's electromagnetic energy coming very firmly into my eye. It's also bouncing into your eyes, right? You're hearing my voice. There's acoustic energy. That's longitudinal wave. And then we have the transverse wave for the light energy. There's energy out here. We transduce it and we turn it into action potentials. At the point that we capture that photon, right, at the point of, and we'll come to the cis-trans isomerization in a moment, and when we first grab that, we've lost all information about wavelength, right? There's some wavelength information out here in the real world, but when we catch it, we basically lose it all. And the reason for that is that any one of these photoreceptors is kind of like a one-trick pony. It either catches the light or it doesn't catch the light. Okay? The other day, I bounced that wiffle ball off the back of this, the back of the wall, and Jake was able to catch it, and so he caught that one. He was basically went through his cis-trans isomerization. We'll unpack that in just a moment. He caught that light. Then Jake would begin to send a signal to the next neuron in the chain, right? The next neuron in the sequence of events. But at that point, it doesn't really matter whether he caught a long wavelength photon or a short wavelength photon. He's a one-trick pony. He says, I got one. And he passes that information down to the next. He doesn't say what kind of one that was. He doesn't say it was long wave. He doesn't say it was short wave. He doesn't say it was medium wave. He just says, I got one. And he passes that down to the next neuron, to the next neuron, to the next neuron. So by the time he catches it, at that point, we can no longer say what kind of wavelength that was. It's univariant. Who's following that? Yeah? Some people, this sometimes helps. Imagine that you've got a pet dog, and the dog is looking out your front door, okay? And the dog sees maybe a bird and starts barking at the bird. The dog is responding to the bird. The dog might also bark at a cat that goes by. The dog might also bark at the mailman as the mailman goes by. Okay? You get a bark when there's something out there. In most cases, you can't tell by the bark of your dog exactly what is out there. You know you caught something, and there's something there, but we don't really know exactly what it is. Same thing with a photoreceptor catching a photon. That is, it can say, I got one, but it's actually lost specificity about what that is. And the way that we try to recover specificity, of course, is we have different kinds of photoreceptors that are maximally tuned to maybe 440 nanometers, or 530 nanometers, or 560 nanometers. We can say probabilistically that particular cell probably caught something on the order of 440 nanometers, but we can't really be sure. It could have caught a wavelength much longer or shorter than that. It's a univariate response. It makes the response or not at all. That's all we get. Okay, so that's univariance. Okay, can somebody tell us a little bit about the cis-trans isomerization? It's a big word. It comes to us from biochemistry. We all did the gesture for cis-trans isomerization. Meg's got it. Can we all do the gesture? Okay. Maybe people have heard about cis-trans isomerizations in biochem classes or biology classes. Yeah? Does it sound familiar? Okay. Do you remember a lot about it or not so much about it?
I mean, is it where you, like, especially like a double bond, if there's two substitutions on the cis, they're on the same side, and then they get switched to the opposite side.
Excellent. Excellent. Yes. Okay. So that's actually more information than what we needed, but the idea is that we get a change in form of this molecule, this Jake, if you will, that's going to catch the photon. Okay? It's in one particular form. When it catches the photon, it changes to this other form. Here's the cis, here's the trans. And I'm fascinated that they can determine that the retinal, which is the particular substance that does this catching for us, right? This pigment that we have, they can tell you that the cis-trans change happens at the 11th carbon atom. There's this long carbon chain, very interesting, and they can tell you that it happens at the 11th carbon atom, which I think is really, really cool. Really cool, so that they have that level of precision. Okay. So when that happens, that's basically Jake catching the photon. We've lost all that wavelength information. Okay? And by the way, what we have to do is regenerate another pigment, another molecule retinal that can be in this form to catch the next photon. So your eyes are continuously replenishing that. It's why carrots are good. Carrots are filled with vitamin A, and retinal is a derivative of vitamin A. So you're doing your eyes a favor when you're taking in any kind of vitamin A, and carrots are really, really loaded with that stuff. Okay, so that's our idea of a cis-trans isomerization, a change of shape. Okay? That's all that is. Okay, then we got to this other weird acronym that I made up called RH Bagels. Anybody remember hearing about RH Bagels? That bagel store? What does RH Bagels stand for? Generally, what is it telling us, the RH Bagels? Yeah, thanks, Alex.
It's from R to S. It's the pathway that I guess electromagnetic information and the signal follows.
Yeah, right. Yeah. Okay, right. So exactly, how do we get from the cis-trans isomerization all the way back to the cortex? We have a sequence of events, and we can say that the very first stop there is the retina, and that's where the photon is caught, right? So we'll say that R is for retina. How about the H? Does anybody recall what H stands for? Okay, got it? Okay, the horizontal cells. And there's a ton of machinery inside of your retina. What I'm giving you in RH Bagels is grossly simplified, but plenty for our purposes. And one other point I make in the video several times is that all of that highly complicated machinery takes place in a region of space that's thinner than that of a postage stamp. And really, really cool that all this goes on. But we catch the photon with the cis-trans isomerization at the retina. The photoreceptors do that. Then we go out to the horizontal cells. Then the B is for, so I'll yell it out, bipolar. Jess has it. Okay, the bipolar cells. A is for amacrine cells. G is for ganglion cells. Ganglion cells are very interesting and very important for us. Their axons form the second cranial nerve, which is going to be transmitting all that information way, way back. And it's that bundle of axons from the ganglion cells that actually makes the optic disc that we talked about a moment ago that gives rise to the perceptual phenomena of the blind spot. Okay, so those are the ganglion cells, and they have the on-center variety, the off-center variety, which almost everybody in the room very nicely schematized through draw-to, so thank you for that. Okay? So after the ganglion cells, I've kind of given it away. The O stands for optic nerve, okay, and then the optic nerve makes its way eventually to the lateral geniculate nucleus, a portion of the thalamus, okay, that's relatively lateral as opposed to medial. So there's one on the left, there's one on the right. You'll sometimes hear me say one of your LGNs, meaning either your left LGN or your right LGN. I think that phrase comes up in the TED Ed questions for Monday. I'll say, in a given LGN, and what I mean by that is either on the left side or the right side. They're left and right parallel structures there. In this relay station, that takes us off to the S in R.H. Bagels, and the S stands for the striate cortex. The occipital lobe contains this primary visual cortex, which goes by many names. Area V1, Area 17. V1 is for primary visual cortex, striate cortex. So that gives you some idea about how we get from one to the next. Okay, I do want to get off to the other questions that we had today, but one more, just to make sure we're all on track. I think this is especially important when we're thinking about ganglion cells. So Jake has already done his thing. He got the photon. He's got the chain going, right? We're going to get something, eventually, some information to our ganglion cells. And there we have this on-center, off-center kind of idea going on. And it raises the idea of the Michelson contrast formula. Can somebody tell us what that is, either computationally or conceptually? The Michelson contrast formula. Okay, thanks, Meg.
Is equal to, in the numerator, the difference of the maximum luminance level and the minimum luminance level
Okay.
over the sum of the maximum luminance level and the minimum.
Okay, it's a fairly simple formula and a very helpful one. Right? We can think of this simply as the difference between the highest level of light. So on this particular screen, a ton of photons are coming off of here. Fewer photons are coming off of there. If we pretend that's the darkest point on this screen, I can put my photometer right there. I can count the number of photons, and I'll get some level. And because that's the smallest level, that'll be L min, the minimum luminance. Right? One of these white regions will be a maximum region, and I can find out what kind of a difference do I have between the most luminous and the least luminous, L max versus L min. Okay? I get that difference score, and then here's the difference, here's the sum. I put the difference over the sum, and I get some proportion that tells me just how much contrast there is. So if this were entirely uniform, the whole thing were white looking, then the L min would be exactly the same thing as the L max, and we'd have zero contrast there. Right? Okay? Who's following that? Is that working for us? Okay, so this will range between 0 and 100, if you'd like to think of it that way, if you think in proportion 0 to 1. Okay? Right. And you might remember, too, that we had that nice example that J.J. Gibson had given us, and that is that sometimes we can tell if we're in a sauna and the fog is really, really steep, we can tell that it's bright or it's dark, but we can't see our hand. We can't see where L max is and where L min is. It's all the same. So our photoreceptors are doing their thing. The Jakes are catching all those photons for us, but there's essentially no relevant information. We wouldn't know where to step. We wouldn't know where to reach. There's no reduction of uncertainty. So the photoreceptors are grabbing the light for us, but we really need some give and take between all these on-center and off-center cells to tell us where are the borders that we can step on, that we can reach to, and so forth. Okay? And all of that still is happening inside of your retina, even though that latter part, where this becomes important, is happening a few synapses past the point of transduction, where we have the cis-trans isomerization. Okay? Anything else? We'll take a moment we made our way all the way through this from the other day. We've talked about the human eye. Okay. Here's the principle of univariance, RH bagels. You've already done some nice on-center and off-center cells. There's L max, L min. High contrast stimuli, moderate contrast, no contrast. Right? Any questions on any of that? Okay, why don't we go ahead and go to today's main event, or one of two main events for today, because at 1:05, and I hope you'll stop me in case we're midstream here, poor Rachel will be waiting for us in Houston, and we need to check in with Rachel at 1:05 and hear all about optometry school and what she's doing with the human eye. I think that'll be really interesting. So why don't we go to the blind watchmaker questions? We had two items that were written for us by Richard Dawkins today, and some of the questions were from the Blind Watchmaker on explaining the improbable, chapter 1. And then we also had a really interesting chapter that came from his book called The Greatest Show on Earth, and we'll spend some time on that. What I think I'd like to do is just go through the first 6 in this format, and then we'll let you have a pair share on number 7, because that's where it gets a little bit, I think we have a lot of room for creativity there, and I want to make sure everybody gets a chance to speak with a partner about what they came up with there. So I wonder if somebody can just start us out on question 1, which was what Dawkins means by hierarchical reductionism. Tawny's got something.
So it's like explaining things in simplest terms.
Okay.
To the math. So if yours, one definition to explain something that explains something else.
Okay, all right, so we do want to be able to reduce. The word reductionism is there, right, and we want to be able to state things in terms of its parts, okay? It's one of the ways of thinking about reductionism. What I didn't quite hear in your response, I heard a nice definition of reductionism. Can somebody help us out with what we mean by hierarchical? So Tanya's got us on a good start. We're talking about explaining something by reducing it to its simpler components, right? But why do we say hierarchical? Well, what's the emphasis there? Alexa's got something.
Sort of in terms of like levels of complexity.
Okay.
Things should be explained at the next level down.
At the next level down, not all the way down, right? Okay. And so some people are critical of the idea of reductionism because when we go all the way down, we're down at things like quarks, right? So we're all made out of quarks. And this is made out of quarks, and you're made out of quarks, so what's the difference between you and this thing? Okay? Well, that's not really helpful. You're very different from this screen, right? So what we want to do is try to understand things maybe one level down. And if somehow that doesn't satisfy us, we can go down one. We don't need to go all the way down, and we in fact shouldn't go all the way down. We go down to, if you will, one lower level in the hierarchy. So between Tawny and Alexa, I think we've got there that we want to be able to explain something by reducing it, but only one level at a time. There are people who are critical of that. I know that there are folks who will say that, well, then you miss what's going on in the bigger picture. You might imagine that the Gestaltists would be very interested in seeing what's going on in a wider context, not a more reduced context. And that's fair. I think that's a perfectly fair explanation. But many scientists do engage in this type of reductionism, hierarchical reductionism, and it can be helpful in explaining certain phenomena. Okay. Let's see. Why don't we? I think we have some idea about how we relate hierarchical reductionism to the study of sensation and perception. Can we get to number 3? Dawkins distinguishes between things that are complicated and things that are not. What are some of his criteria? What's about complicated and not so complicated? Why don't we go with Abby and then with Alex?
They're complicated or like heterogeneous.
They're heterogeneous.
I mean, they're not the same.
They're not the same on each side, right? So what does he call it? Blanc mange or something like that. Right, and I don't even know what that is. Is that a pudding or is that? Okay, yeah. I've never met a student who knows what that phrase is, but we all seem to get. It sounds like it's a pudding. So you can have a bowl of pudding. You can cut your bowl of pudding in half, and the left pudding is the same thing as the right pudding, right? Okay. But if you were to transect your eyeball, okay, well, the anterior portion, which has such things as cornea and lenses and pupils and so forth, is very different from the back half, the posterior half, which has such things as photocells. Okay, so we get the idea that cutting an eye in half is really not the same as cutting pudding in half. Okay, so one of the ideas about what is complex is there's a lot of heterogeneity. There are some other criteria too. Somebody want to offer some other criteria? We'll go with Alex and then with Jess.
It has to have some quality specifiable in advance that is highly unlikely to be acquired by random chance.
Okay, so some principle, highly unlikely that we can specify in advance, that it probably didn't happen just by chance, okay? Was that the one that you were going to offer? Okay, that's a lot, right? That's a lot, because what you could say is if you take now, here's another one, Mount Blanc. Who's heard of that one before? Mount Blanc. Is that a peak in the Alps? Is that what that is? It's a mountain anyway, but I don't know where it is. I don't know where, I should know where Mount Blanc is, but maybe they make pudding there or something. In any case, you can cut that, and the two sides aren't exactly the same, right? They're not exactly the same, so they're a little bit different, but it has to, something about the heterogeneity that Abby pointed out has to be able to do something for us. And what he comes back to is, well, it has to be able to do something like earn a living in some way. And some things earn a living by phototransduction. That's what plants do. Other things earn a living by swimming, some by flying, some by digging. Okay? We can specify in advance what that feature might be. And so it's unlikely that this would occur just by chance, that you would find a flying organism just by chance, or something that, that sings as well as particular kinds of birds sing. Okay? Okay, let's see. I wonder if we might go to, why don't we just jump right into number 7, because I do want to reserve some time. So can you find a partner? And I wonder if you can share with your partner one idea from the Blind Watchmaker, right? One idea that you think is relatable to something that you've learned in some other course taken at Denison. Okay? So can you find a partner and share something from this chapter that you related, and then we'll come back as a group and see what we've got. Find a partner. Be in a trio if you need to be in a trio.
Right, I
I had
Yeah, it's all like, well,
can't have one without the other, but it's basically broken down.
Okay, I don't know if everybody got a chance to share their idea, but hopefully you at least got to exchange some ideas. Somebody want to share with us either what they thought of or what their partner thought of? Let's hear about a connection or two. Okay, thanks, Alex. Alex has the hot hand today.
I did structuralism and synthesis.
And synthesis, okay.
Which is, the breaking down parts of the mind into simple components, and then synthesis, seeing how they work together. Kind of reminded me of hierarchical reductionism.
Okay.
Breaking things down into complex things down until we don't have any, I guess, any questions to ask, and seeing how those parts interact.
Okay, all right, that certainly works. Somebody else want to share what they had in their response to number 7? Okay, thanks, Sarah.
I was about the importance of definition.
Ah.
And then we talked about the differences like complex, and then in philosophy we do a lot of like defining the terms so that we understand beforehand what we're trying to get into.
Okay.
Because like I thought it was interesting like included like computers as like biological objects.
That was very interesting, right? So according to his definition of a biological object, a computer satisfies the definition, right? So there's this notion of how do we define something, and then what kinds of objects will land in accordance with that definition. Okay, so he's kind of stuck with computers, and at some point I think he says something like, well, I don't care whether computers are really biological objects or not. Words are our servants, not our masters, and we need to use them as we need to use them. Right? Okay? Okay. Something else? Maybe we can get one more? Okay, thanks, Zach.
Ecology and evolution.
Okay.
So just learn ways that like organisms adapt to their environment, and the way they evolve would fit the definition of complexity because it's not, it has like a prescribed purpose. It's
okay.
not just entirely random.
Okay. Mm-hmm. Yeah, certainly. So there's a nice connection to something being complex, and it has to serve some kind of purpose that we can specify in advance, and you're probably all over that in ecology and evolution. That it's really going to be all about that. Okay? All right. Anybody else dying to get in? We can take one more if we need to, or we could move on. Okay, why don't we move on, and why don't we move on in this way? We have about 7 minutes before we get to our interaction with Rachel, so why don't we divide and conquer? So if you're over on, why don't I make the dividing line over here? If you're over on this side of the room, can we move on to the greatest show on earth, and we'll look at question number 1, and we'll ask the people on this side of the room to address question number 1. Maybe you can chat with each other about the gist of these experiments that were going on in Lenski's lab. They're actually, I think, very beautiful. You can tell from Dawkins' writing that Dawkins is very impressed by these experiments. And they're a little bit complicated, so maybe you can share with each other your summary of that gist. And then I wonder if we can have this half of the room talk about question 2, the second one that we had for the greatest show on earth. And this was that experiment that was described on pages 126 through 130, and they were talking about the issue of irreducible complexity and how Lenski's experiments may have addressed irreducible complexity. So why don't we let you chat on your question with your partner for a couple of minutes, then we'll share what we've got, and then it'll be time to check in with Rachel. So go ahead.
That part.
I'm always tired.
Okay, we've got about 3 minutes before Rachel comes on board. So why don't we see what we get? Maybe we'll make it through at least one of these. And if not, I hope you'll hold those thoughts until Monday. We can pick up on that. But I would like to get a feel for what's going on in these Lenski experiments. You could experience Dawkins' excitement about these. He thought these were really terrific. So can we have somebody maybe from this half of the room get us started anyway? It can be a team effort on what's going on, the gist of Lenski's experiments. There's a lot to explain there, so we can kind of start where we need to, and we can go backward or forward. Thank you, Gabby.
Okay, so Lenski and his team had 12 flasks of all the same E. coli
over the course of 20 years, this team had them grow and eat and evolve separately into different strains. And so the food they were given was glucose and a mixture of a bunch of other things. And glucose was the limiting factor, so they were hoping that all the bacteria, the E. coli, would adapt similarly to use glucose efficiently. And so one of the strains was that one of interest in it used citrate, and it mutated to eat citrate instead of the water and the food. But they became able to do that, and they didn't expect that.
Okay.
It was very informal.
Okay, very good. So nicely laid out. We've got these E. coli flasks, and there are 12 of them. You might think of them as 12 founding tribes. They use words to that effect. And every day, they take a little splash from one of them, and they put it into a new flask, and now they have day 2. And then they have day 3, and they do this for 20 years, and they've got tens of thousands of generations. I can't imagine how these flasks are stored. That must be a tremendous amount of space. Of course, any one is not particularly large, I'm sure. But still, to have 20 years of flasks on 12 different tribes, right? And we're looking at mutation rates. We can measure such things as optical density. We can see how well they're doing with their splash of glucose and so forth. And then all of a sudden we get these mutations that come out of nowhere. And that's going to be what this group's talking about. But how many people followed that general gist of his experiments, right? And I believe they're still ongoing. Okay? All right. So I would like to hear about how that line of experiments deals with the important issue of irreducible complexity, which is a point that is frequently raised by people who really don't believe in evolution. They say, well, how can something as complex as the eye come on board simply by random mutation and non-random selection? There must be something else going on there. And Lenski kind of has a counter-argument to that this group will help us out with maybe on Monday after we come back to that question. But we're at 1:05 now, and I promised Rachel I'd chime in at that time. Why don't we see if we can get her? Hey, Rachel. Hey, can you hear me? Okay. I'm having just a little bit of trouble hearing you. I'm going to crank you up just a tad. Okay? Oh, that's quite all right. And can you see me? You can't see me. I'm sorry. Let's see. Hold on. I don't know if that's going to do anything. No, I don't want to turn the video off. Okay, anyway, you can, okay, so you can't, okay, that's great. Well, anyway, we can all see you. We're projecting you on the screen. Hey, we're projecting you on the screen where you had S&P back in the spring of 2013. Rachel was in our founding class of videos. So Rachel was in the first class that had the videos that you've all been seeing. And I think, it's a shame you can't see because you might have some familiar faces. Do people know Rachel? So you were first year when Rachel was a senior, maybe? So Meg is with us. Anybody else know Rachel from their first year? Meg's waving to you anyway.
Hi, Rachel.
Okay, if I can figure out how to turn that our video on. Okay. Well, anyway, just in the interest of time, we have only 15 minutes, and you're a very busy optometry student. So what can you tell us about life after Denison or optometry school or anything else you'd like to tell us?
Yeah, so is anyone in the room interested? Do you have any interested optometrists or anyone interested in entering the medical field or vision related field at all?
Maybe medicine. I think maybe medicine, yeah.
Okay, yes, I'm here to speak to how optometry differs and what the process is like, because the admission process is really similar, and I just went through that this past year. So after Denison, I worked in optometry and then an ophthalmology office, and that was a really great experience for me just to figure out what I wanted to do. Does anyone know the difference between optometry and ophthalmology?
So I'll be the translator for us. Anybody familiar with that? Let's do a show of hands. How many people are familiar with the distinction between optometry and ophthalmology? Okay, Alexa, kind of. Do you want to tell us, Alexa, what you know about that?
Is optometry like corrective, that sort of stuff, and then ophthalmology is like, I don't know,
she's pointing to
surgery on the eye or something?
Yeah. Okay.
That's exactly it. So if any of you are interested in entering medical school, that's a great option. And both careers deal with correcting vision just in different capacities. So it's good to know the differences and know what you want to do. And just because optometrists don't do surgery We still do a lot of important things. Lots of times we are one of the first doctors to diagnose people with stroke, diabetes, blood pressure, and really tell a lot about general health on the eye. So lots of important stuff there. And as far as going to speak a little bit about the admissions process, kind of going through that.
That'd be wonderful. And if I can just interrupt on that, you made an important point that when doctors look at the eye, even if it's just they've got this scope that can look into your eye, it's a handheld scope, and they're getting a good look at it. I've been told, and you can tell me if my friend was putting me on. One of my good buddies from graduate school would look into eyes, and he said he could tell whether there were relatively high or low levels of cholesterol by looking at your retina. I don't know if you've gotten that far yet or not.
Yeah, I haven't heard that. I know I hear a lot about blood pressure and diabetes, but I'm sure, I mean, like I said, the eye is predicted a lot, but I'll have to ask one of my professors on that.
Okay, all right. So anyway, you were about to tell us about the application process.
Yes.
Okay.
Yes. So for anyone interested in medical school or dental school, optometry school, it's all a pretty similar route, so I can just go through that. And for anyone, if you have questions throughout or if you want to email me afterwards, feel free to. I'd love to be a resource for you. I think it's helpful to hear from recent students just on their experience if it's fresh in our minds. So feel free to interrupt me at any point. But as far as entering school, entering graduate school, first you have to take a few prerequisites. So it's similar to medical school. You have to take classes in physics, chemistry, biology. Each school has pretty much the same requirements. A few need like an additional anatomy or something like that. But you can find all that on their website. So if you're working on that in undergrad, that's great. If you still need to take a few classes when you finish, that's fine too. I know some of my friends took summer classes. I took classes the year after I was finished. So that's always an option. And then the next thing is shadowing. So whatever you're interested in, I found it to be extremely helpful. Dr. Matthews actually connected me with an optometrist in Newark, and she taught me a ton. Just seeing what she does day in, day out, some of the volunteer trips that she does, that was really inspirational and really helped confirm my decision to enter optometry school. So I highly recommend that. It's also required for a lot of schools to have some check experience. So it's good to do that as soon as you can, see if you're interested in it. And then from there, once you're interested in actually applying, you have to take an entrance exam. So for optometry school, it's the optometry admissions test. For med school, it's the MCAT. In dental school, it's a different test. So that takes a little bit of studying, but hopefully you'll all do well on that. You will from Denison. And then after that, you kind of start, it's similar to like the Common App. You use the Common App to apply to undergrad schools. So it's an online application process, and you enter in all your transcripts into there. You have letters of recommendation filled through there. One of them has to be from an optometrist, and it's really helpful to have shadowed someone, create that relationship, so they can write a recommendation letter for you. And then from there, you submit that application online. And then you send it off to however many schools you want. So there's only 20 schools in the country. So not every state has one. But there's, they're kind of few and far between. So it's interesting to see where they're located. So after that, then you hear back, you're invited to interview, and the whole process takes at least, I'd say maybe a month and a half on the short end, but it's up to 2 months for everything to be finalized, and you're back from school, and at the interview you really get a good idea of what the schools are like, and then it's a rolling admissions process. So it opens up in July, it closes in March, so it's a pretty wide time frame to get all your stuff in order. So that's it's similar to, you all apply to undergrad schools, it's really similar to that. You just require a few optometry specific essays, and you have to show general interest in the field. So again, shadowing is really important there.
And if I can interrupt you there, Rachel, so that's really a very helpful overview. If students have questions, please raise your hand and I'll relay the question over to Rachel. But I did want to say one thing. She mentioned that when she was here at Denison, she spent some time volunteering with a local optometrist who is stationed in Newark. That optometrist, she's been to this classroom actually. Her name is Janine Flood. Right? And I think you weren't in the semester that she visited. I think she visited a semester before you.
I think so.
Yeah. her name is Janine Flood. If you're on North Pearl Street, and there's a cross street there called College Street, the one house that has all those gorgeous flowers and this light purple kind of shutters, that's Janine's house. And I was out on my run yesterday, and I told her, Rachel, that we would be Skyping today, and she melted. She wanted to send her best wishes to you, and she was so happy to hear that you're on optometry school. So, yeah, right.
Yeah, I think she was one of the very first optometrists that I ever shadowed. So
Yeah.
it's
Yeah.
exciting, and she was great to talk to during this first show.
So,
Yeah,
so
she's a great resource for everyone.
So one other thing I'd like to share with the group, and that is that I'll embarrass Rachel here, but Rachel was an outstanding student, as you might imagine, and she got into a lot of different, at least 2 different optometry schools, including Ohio State, which has a terrific program, and Houston. And I can imagine that once you really get into shopping for these things, those are both really strong schools, but maybe one of them had a topic that was more of interest to you. And so there are different ways of maybe specializing within optometry or ophthalmology. Can you share with us maybe some of the distinguishing factors that make one school more attractive or less attractive to different kinds of students?
Sure, yeah. So that's something that I didn't really realize until I started looking at schools, is that you can really do so much with optometry. You can go into general practice, and go over in practice, if you're interested in the business side of things, that's a great option. And that's something that Ohio State really emphasizes, is they emphasize they call it their practice management. So they have a really strong program there. Ohio State also specializes in contact lenses. So if you want to specialize in that, mainly if you want that to be kind of your main population or people with contacts, then that's a great deal too. Here at Houston, we have a neuro-optometry department. So that was something I was really interested in. So they deal a lot with, tons of different diseases. We have doctors from all over who specialize. So I think that I'm interested in disease and maybe low vision work, which we also have a low vision office here.
Okay.
And those are kind of up and coming fields of optometry.
Yeah.
We work a lot alongside ophthalmologists, dealing with work there. So that's something that really drew me here. It's the whole center for low vision work, for neuro work, disease-related work. I think there's just a ton you can do with that. So that was something that drew me here. And then also there's a surgery center here. So if you want to help ophthalmologists, they would do like the LASIK surgery, but if you want to see patients before and after, it's really great practice here since we have the surgery center right on site. So that was something that drew me here too.
Wow.
Yeah, so lots of different options. There's a lot you can do with it. You can, that was something that's important, is that you can be flexible. You can work 2 days a week. You can work at a retail office. You could work at a hospital, full-time. So really there's a lot of variability, which is always nice.
Yeah, that's great that you have that level of flexibility. Let me scan the students and see if we have any questions.
Yeah.
At least, maybe not at the moment, but I did want to pick up on your comment that Houston is really tightly tied to their neuro program. And I know a neuroscientist, vision scientist there, whose name is Scott Stevenson, and you probably haven't. You know Scott?
Yeah, he's one of my professors.
Oh my goodness. Okay, right. Yeah, that's how I know Scott, is because he actually finished at Brown a few years before me, but his picture was on the wall, and it was a big inspiration for all of the PhD students to say, okay, you're going to be next on the wall. So I'm 1 or 2 rows over from Scott on that wall at Brown. But I'm glad you've made the connection already. He is a neuroscientist.
Yeah.
So what I also wanted to ask about is when my other friend, the guy who was telling me he could see the cholesterol in my eye, he told me I had very low cholesterol levels from my eye. He also told me that people had very diverse backgrounds. You were a psychology major.
Right.
Some people, and you were relatively right out of school. You might have been off for a year or 2, but there are other people who are coming into his program who are 45 or 50 years old, right? And they had been lawyers, and now they're going to optometry school. I bet you see a range of training coming.
Yeah. I have, there's someone here who just finished their master's. They're in my class. They just finished their master's in business, and their whole plan is they want to start their own practice, and they want that to be their primary goal. There's someone else here who majored in electrical engineering. So that's an interesting major coming in. There's a lot of biology majors, but also we have some art majors. You can really, people have a variety of interests. It's really nice, meeting people with different backgrounds. And like I said, as long as you take those prerequisite classes, you can really major in anything else. So I thought I would opt all out in a psychology major, but I actually found that my neuroscience classes, one of my classmates didn't have a really strong neuroscience background.
Okay.
I'm going a whole neuroanatomy class, and then my optics class that Dr. Stevenson is the professor for, that's a lot of what we learn in sensation and perception. So a lot of that is kind of overview of what I've had before. So it's always nice when something's reviewed rather than great.
Great. Something else. Do our students have a question or a comment? That was a stretch? Okay.
The stretch, yeah.
No, there was somebody was stretching.
I can tell you about the process. So the 4 years, all of them are
Please.
different.
Yeah.
I didn't know about this before I entered, but your first year, I'm primarily in classroom. I still have labs this semester. You take about 20 credit hours, and just a wide variety of classes. I'm in an optics class, which is physics-based. Dr. Stevenson teaches a vision science class. We have general optometry, we have physiology, neuroanatomy, anatomy. I think that's most all of them. So first year is just kind of getting your solid coursework down, making sure everyone's on the same page. And then your second year is really exciting because when you start to see patients. So kind of putting everything you learned first year into practice. So you do about half your time with patients and half your time still in the classroom, learning, becoming more specialized in learning. And then your third year, you're primarily with patients, is really trying to fine-tune all your skills. And then you still have a few upper-level classes. And then your fourth year is really exciting. They're called rotations. So you do 3 rotations. So each one is a few months long. And you can do those forever. So I'm based in Houston, but I could go out and do a rotation in Colorado. I could do one in New York. I could go to Florida. Basically wherever you want. And there you really start to specialize. So if I wanted to work with low vision patients, I could work, in a hospital setting or a VA or somewhere else that specializes with that. If I wanted to do surgery, like work in a surgery center, I can find somewhere that specializes in that. If I wanted to do pediatrics or work with the cornea and contact lenses, there's so many things, and so that's when you really start to narrow in on what you're interested in. So that's your fourth year, and you go to 3 different places just to get a variety of views of what kind of places you would want to work in. Maybe there's somewhere that you wouldn't like as much. So that's something that everyone always looks forward to, is the place in my fourth year and getting to explore somewhere new and really get into working right alongside another optometrist, learning from them. So
that sounds great. You've given us a 4-year overview in just a couple of minutes, and we appreciate that.
Yeah.
I wonder if I can ask this group, can you please join me in thanking Rachel for spending some time with us today?
Thank you.
All right.
If anyone has any questions about entering optometry school or medical school in general, I'm happy to, feel free to reach out to me. I'm happy to answer anything.
Okay. Rachel, thanks a million. Thanks a million. Thanks so much.
Thanks, everyone.
Okay. Bye-bye. Wow, pretty cool. Well, I hope you'll be Skyping in a couple of years. Whatever it is that you're doing, we'll find some kind of S&P connection. I look forward to seeing you on Monday. We'll finish up our conversation on Lenski, and we'll also have some new TED Eds to work with. Okay? Have a great weekend, everybody. Get some REM. Oh, no. Do you have to get up at 8?
No, my body's just programmed.
Yeah.
Oh, you just do that?
You just wake up no matter what time I go to bed.
Isn't that funny?